In this lecture, we will discuss about the stability of proteins upon amino acid substitutions. In the previous lecture, we discussed about identifying the stabilizing residues in protein structures. For example, if you know a structure, which residues are important for the stability. So, we used various criteria like hydrophobicity or long range contacts and conservation to identify stabilizing residues, right, in protein structures. In the later part, we discussed about the database, a thermodynamic database for proteins and mutants, the ProTherm. It contains the data for the proteins as well as mutants, right, obtained from different experimental techniques like sectoral dichroism or differential scanning calorimetry, fluorescence spectroscopy and so on. So, here now we will see what will happen if we mutate a specific amino acid residue in a protein. Here you see there is a central residue here, so it is a central residue. If we mutate this particular residue with any other residues, currently this residue is making contacts with other residues in the protein. If we mutate this residue, it may lose the contact or may make additional contacts, right, depending upon the type of mutations and where you mutate, either depending on the secondary structures or the location based on accessibility. So, here I show some types of amino acid residues, right. Here you can see the positive charge residues lysine, arginine and aliphatic residues. If we mutate alanine to valine, what will happen? If we see alanine here, if we mutate this alanine to valine, so whether it stabilize or destabilize, or this will make contacts or it will make a steric, right. So, that depends upon the location where you mutate, right. Likewise, if we mutate alanine to lysine, what will happen? This will stabilize the protein or destabilize the protein? Destabilize the protein if this is in the buried state. If it is located in the exposed and there is a nearby residue called say aspartic acid, glutamic acid, in this case that can make salt beds, right, fine. So, if you see this mutation, if you mutate alanine to valine, either this will make a collision, steric hindrance, or it can well packed depending upon where the alanine is located and how far space is available, right, nearby this residue. Likewise, if we mutate valine to alanine, it can create a cavity by destabilizing protein or you can freely move. So, if you say alanine to aspartic acid, either make the electrostatic interactions, right, or may destabilize, right. Likewise, lysine to leucine. So, if you mutate a specific residue in a protein, so it may alter the structure or alter stability as well as alter the function, right. Some of them may also lead to diseases. For example, the hemoglobin, if we mutated glutamic acid 6 to valine, it causes sickle cell anemia. Like if we say P53, right, it is a DNA binding protein. If you make a mutation, specifically origin mutation, right, this will lose the binding affinity. Finally, it may lead to certain types of cancers. Now, I show some examples. So, there is a negative charge residue and you have positive charge residues. Right. If it is close to each other, then they can make ion pairs. If you mutate this positive charge to negative charge, right, if you mutate this one, then what will happen? Here these two residues are negative charge. So, they make the repulsive interaction. So, in this case they may destabilize the particular protein. The second example, if there are two residues and this is small residue. So, big residues, this is not representing charge, this is a small two big residues and this is small residue. For example, here to valine, right, and here you can see this alanine. If we mutate this alanine to leucine, what will happen? Here if you see lot of space. So, because we mutate alanine to leucine, which one is bigger one? Leucine, right. Here we put leucine here, right, here is valine. So, this occupy all the space, this is a cavity filling mutation and this is more compact. In this case, this will stabilize the protein. On the other hand, if there are two valines here and the alanine is here, the same if you mutate alanine to leucine A to L, this is L now, then this are V. So, what will happen? There is no space for leucine to accommodate here, because it is very less space where only alanine can accommodate, and we mutate alanine to leucine. So, here this will create steric hindrance and this will destabilize the particular protein. So, the stability of a protein upon mutation, it depends on the location and which residues you mutate, right, as well as where you mutate that particular residue. So, how to understand, right, the factors which influence the stability. In this case we use the database, right, which database contains the data? Protham, right, Protham contains the data. It has more than 25,000 entries and more than 12,000 or 13,000 single mutants. So, now we can search this database based on various search options available. For example, you can see the mutation single and you can see different types of other options available in the database, and finally, you will get the data. Once we get the results, then you can download the data and utilize the data to understand the factors which can stabilize or destabilize particular protein. So, how to understand, how to estimate the contribution of different properties, which property can relate the stability. Protham gives you experimental data, right, either you can get the delta Tm or delta energy. This is the transition temperature, the melting temperature, and delta energy you can get this free energy change. Right. So, delta G you can get this equal to delta G mutant minus delta G wild. Experimental data can provide the delta G values for the wild type and for the mutant, and you can calculate delta G using the equation, right, delta G mutant minus delta G wild, right, this is known. Like delta Tm also you can calculate if the experimental data are available. Then the other side, so to which properties you want to relate, right, because we know there are several behavior of these amino acid residues. What are the characteristic features of different amino acid residues? You can polarity, hydrophobicity, right, alpha helical forming tendency, right, the free energy, right, various properties. So, on the other hand, you can take the different properties. The properties you take from different perspectives. For example, size. There are several physical properties. For example, molecular weight. In this case, you can see the glycine has less weight than the other residues, and you can see the bulkiness. There are some residues which are very bulk compared to other residues, right, some are loose and some of them are very bulk, and you get the shape and the volume, refractive index and so on. There are many properties, right, you can deal with the physical behavior. There are several chemical properties, right, for example, hydrophobicity and polarity, isoelectric point, right, several chemical properties. There are several energetic properties, short range interaction energy, how the two residues are in contact with each other based on short range or medium range or long range, the frequency of occurrence of these residues compared with the all the residues within that range, you can convert this into potentials, and from this one you can get the energy. So, like say long range non bond energy, right, you can see for each residue how it can make with other residues. Then you can get enthalpy, entropy, free energy and so on. These are the properties you can calculate for each residue, right. For example, if you have the molecular weight, you have 20 values for the 20 residues, right, for the volume for 20 residues you have 20 values. Likewise, you have the conformation properties, right. How to get the conformation properties?
From known structures.
Right, we get the propensity, right. How to get the propensity of alpha helix?
Calculate the probability of the.
Right. For any residue I, you can get the take any residue I in alpha divided by the residue of I, the whole protein, normalized with n alpha divided by n, right. This is any total number of residues in a protein, how many residues in alpha helical conformation, like for any residue I, how many residues in alpha helical conformation and how many residues type I present in the protein. So, you can calculate alpha helical propensity. So, what will you get the propensity, how many values you get? 20 values for the 20 residues, right. Likewise, beta strand you get 20 values, right, everything 20 values. Now, for the property wise, each property you have 20 values for the 20 amino acid residues. Other side you have the experimental data, right. Now, how to relate the experimental data with these properties, like for any property, right, for example, P. So, in this case you can take the difference in properties delta p of I, which is given as p mutant I minus p wild of I. So, for example, this is a p mutant is property of the mutant residue and wild is the property of the wild type residue. For example, you take the shape, shape tells you the branching. For example, alanine there is no branch. For example, if you take the leucine 1 2 1 2, second CH you get the loop the branch. So, in this case shape of this leucine equal to 2, shape of this alanine equal to 0. If A is mutated to leucine, it is not this is leucine, and then A is mutated to leucine. So, delta shape equal to shape of this leucine this equal to 2 minus shape of this alanine that is equal to 0, this equal to 2. Okay, this is the shape of leucine, this is the shape of alanine. So, alanine this equal to 2, this is not so clear. Okay, this is the shape of leucine, this is the shape of alanine. So, the subtracting will get the value 2. For example, take hydrophobicity. For example, you convert this alanine to leucine. Alanine you can say 13.85, leucine if this is equal to 15.20. Then this, for example, if you take the hydrophobicity, what delta Hp this is equal to, right, Hp of leucine minus Hp of alanine. You will get this value, this is equal to what is leucine 15.2 minus 13.85, this is equal to 1.35. So, for any property you can calculate the delta p of I if you know the mutated residue and the wild type residue. So, now if you have 100 mutants, take any particular property, for example, hydrophobicity. For the 100 mutants you can calculate delta Hp, right, you have 1 2 3, right, you know the delta G. You take the hydrophobicity, you have the value of Hp1, Hp2 and so on. Right, if you take the difference, this will be delta Hp. So, you have 100 data for the free energy, 100 data for the properties, and you can take the correlation. This will tell you whether this property is related with the stability or not. So, how to see whether it is related or not? Correlation. What is the range of correlation? It takes from or varies from -1 to +1. If it is +1, that means directly correlated, -1 inversely correlated, if it is 0 no correlation, right. So, it can go from -1 to +1. So, based on these numbers we can tell how far a particular property is correlated with the stability. So, now if you take any type of mutations, for example, some buried mutation. What is buried mutation? Mutations are in the core of the particular protein. So, if we say surround accessibility is less than 5%. If you mutate any specific residue in the core, what are normally occurring residues in the core? Leucine, alanine, valine, hydrophobic residues, right. If you mutate it, right, we will get the free energy already there. So, which properties, right, reflect this stability with good correlation? You can check various properties. Here I showed the 49 properties, and all the data you can get in our website, original values for all the different properties. If you look the 14 properties and relate these 49 properties with the stability, some properties, right, which have good correlation. For example, with the mutation is in the buried, mainly the properties reflecting hydrophobicity, right, they have good correlation with the stability. So, here I show one example, right, x axis is difference in hydrophobicity and y axis is the free energy change, and you can see there are good correlation between hydrophobicity and stability. So, I show another example. Okay, here if you see glutamic acid at position 49 is mutated to different residues, right. What is secondary structure for this mutant? Strand. Okay, this is strand. Where it is located? Buried or exposed? Right, we have the ASA. So, it is 0.4, that is less than 1, right. If you take the less than 5 cutoff, then this is buried. So, because this value varies up to from 0 to 100. So, it is 0.4 less than 1. So, we can take this as the buried one. So, now if we see which properties reflect better correlation with this delta delta G H2O. This is the experimental data and we have several properties. Okay, this is the buried one and a strand mutation. So, we can use the hydrophobicity values to see whether this can have good correlation or not, right. In this case you can draw a graph. Here this is the delta H GM, right, this is hydrophobicity scale, and here you can see delta delta G H2O. For example, we take the first mutation. What is the first mutation? E49G. So, how to get the values? Right, HGM of G minus HGM of E. So, what is G? 13.34 minus what is E? 11.38. What is the value? 1.96. For this case, the value is 1.96, right, 1.96. And the second one, to do this is 2.47 and the third one E49V. So, with this a V minus E, right, this is equal to 3.18, E49I this is equal to 3.90, E49L this 2.75, E49P, how to do this E49P? 12.35 minus 13. No, minus 11.38, right, which is the E, right, this is equal to 0.97 and E49Y 2.50 and 2.55. So, you can get the values. For the last one E49S, serine about 2.01. So, now you plot this as x axis and this as y axis. You can plot first, and if you take 1.96, 1, 2, 3, 4. So, 1.96 and minus 1.7, minus 2, minus 4, 0, 2, 4. Okay. So, in this case, what is the number? 1.96 and minus 1.7, somewhere here. And second one 2.47 minus 0.3, minus 0.3 is somewhere here, right. And the third one, this is E49G, 3.18 is somewhere here, E49V somewhere here. So, you can plot all these 20 values and then if you see there is a good positive correlation, right. Finally, you get a correlation of 0.6 or more or equal or is more than 0.6. So, if you relate the hydrophobicity with the delta G with the mutation is in the strand, right, and it is in the buried region, you can see a good correlation between hydrophobicity and stability. So, at the same time, if you have another set of mutations, if the mutant is not at the buried, right, you can see here. So, the mutation is at the exposed, right, this is the Y20 tyrosine 26, right, here this is the tyrosine 26 exposed, this is in coil. Then what will happen? Here if you mutate this residue, can we see the same trend that hydrophobicity has direct correlation with stability, right. Let us see. So, this is the data. So, here if you see the hydrophobicity scale, so this is the values. Here also I showed that here we tried the HGM, this figure with the HP. So, in this case if you use different scales, you can see similar behavior, positive correlation or the negative correlation. So, here you have these hydrophobicity values and here this is the delta G. So, where this is located? Exposed, because this is ASA, the ASA 76.23 is highly exposed and this is in coil region, and see whether you have any trend with the hydrophobicity. So, here you can make a graph, this is the delta H T, here you can see delta G. Let us calculate the values. Okay, so this is Y26W, what is the value? what is the value of W? 3.77, for the Y 2.67. So, this is equal to 1.1, and the second one 0.2, phenylalanine, what is phenylalanine? 2.87 minus 2.67, this is 0.2, and minus 0.8, minus 0.5, is Q minus 2.67, H minus 1.8, Y26C minus 1.15, Y26D, D is 0.66, so minus 2.01, right. Now, you can make a plot connecting the hydrophobicity and the stability, where you can see that stability is almost, this is fine, this is 0, 1, 2, 3. Here this value is minus 2 up to minus 3, right. This is minus 3, minus 2, minus 1, 0, 1, 2. What is the first one? Y26W 1.1 minus 0.1, somewhere here, and the second one Y26F, this is 0.2 somewhere here, and y axis 0.4 somewhere here, and the third one Y26V minus 0.8, minus 0.8 and 0.9 somewhere here, Y26L minus 0.5 somewhere here, and 1.1, right, this is 1 point somewhere here, Y26Q minus 2.67, 1.4, Y26H minus 1.8 and 1.9, Y26C minus 1.15, 2.2, Y26D minus 2.01 and 2.7, somewhere here. How about the correlation here? You can see inverse correlation, and the correlation coefficient are equal to 0.79. So, if you see the mutations, one is at the surface in the coil region, one is the buried. So, the buried one we can see a direct relationship between the hydrophobicity and stability, but in the case of the coil mutation, look at the surface, right, you can see the inverse relationship between the stability as well as the hydrophobicity, right. In this case, the first time they reported in 1990 that the hydrophobicity has inverse relation with stability, that means increase in hydrophobicity decreases stability for this particular protein, and they published this paper in Nature. Now, the question is, if you see this behavior is universal or not. You can see the same trend, right, everywhere for many mutants, or this only for this one, right. If you see how to do this analysis,
For all the protein.
go to Pratham. For example, here another important aspect is we look at the single mutation. We did not take the mutation from different proteins, right. The same Y26 here we considered. In the last example we considered the E49, this is the location, same protein. So, you can go to the Pratham database and search for the mutations in any single point, whether at least more than 5 to 6 mutants. If it is available, then you take all this information and you relate the different properties. So, we have 49 properties, all the values are available, take each property, get the correlation. Now, if you see most of the cases, if it is exposed, you can see this reverse trend, and if it is buried, and many cases you can see the positive trend. If there is other places, for example, helix or strand or coil, buried, partially buried and exposed, these are 14 properties, and you can see different trends. For example, if it is helical mutation, you may get a good correlation between the stability and helical propensity, or beta strand mutation, this long range interaction energy, there may have good relationship with the beta strand mutations, right. You can do the analysis and you can see very the common behavior to understand the relationship between unique properties as well as with the stability, right. You can easily do with the correlation coefficient. In this mutation we just analyzed the variation of properties, there is only one property, right. Here in this case if we consider only the relationship between the properties and stability, just change the properties and then stability. In many cases it will work, but if you consider many mutants from different proteins, right, you may end up with the very less correlation between any of the properties and the stability. If this is the case, right, for example, same mutation for Y is mutated to leucine, right, same buried, sometimes it may decrease the stability and sometimes it may increase the stability. In this case if we consider only the property, you cannot account the both stabilize and destabilize. For this situation what can we do? So, one aspect is we can get the information regarding neighboring residues. For example, if T is the central residue and T is mutated to valine, here there is another threonine, this is also mutated to valine, right. For example, this T is mutated to valine, this T is mutated to valine, and one place this will destabilize, for example, this place it will stabilize. In this case if you take the property values, any property value of valine minus threonine, you get one value, but the value is the same even if you take here this position or this position. If this destabilize and the second one stabilize, then it is difficult to account both cases. For this case you look to the neighboring residues, for example, in this threonine, right, if you take this T, it is neighboring is R and D, then you put the condition threonine is mutated to valine, but the neighboring residue contains polar residues or charged residues, then this will destabilize. If the threonine is mutated to valine and the neighboring residue is hydrophobic residue, then this will stabilize. So, in this case you can distinguish the mutations, same mutation at different locations depending upon the residues which are neighboring with this particular residue. So, how to account in terms of properties? You can take any window length, for example, we take this and this window length of 3 or here to here window length of 5. So, you can use sigma j equal to I minus k equal to sigma j equal to I plus k, I is the central residue, k is the window length either from left side or at the right side. So, take any window length and get the values. So, in this case you can get the property value for any window length, right, for this you can say window length. This is you can take it as a wild type values. Then you can subtract the mutations, then you can include the sequence information and get this P sequence of I. Now, this P sequence of I is influenced with the property values plus the neighboring residue information, right. When you add this you can account the mutants, right, which are occurring at the different places, right, the same mutations. This is how to include the sequence. You can also include the structure information when you mutate a specific residue. For example, this threonine, right, this is mutated to valine. You can see this threonine is surrounded by different residues. If we take this threonine, right, here you have the different circle, this is surrounded with different residues. You can take into account of the residues which are occurring within the limit of this 8 angstrom and substitute the values, and if you add this, then the value of this T will change from this T. This you can account to the structural information, like say sigma Pj of I, where here I is the any central residue, Pj is a property of the residue J which is surrounded with this residue I. This number will change based on the location of your mutant. In this case, given if you mutate the same residue to the same residue, for example, threonine to valine or alanine to tryptophan, depending upon the location you can get different values, and in this case you can account the stability of these mutations which are having the different positions and similar type of mutations. So, we discussed 3 different types. One aspect is you can directly calculate the change in property and that you can relate with the stability values. If the 2 mutations are same, then we can add sequence information. How to account the sequence information?
Neighboring residues.
Neighboring residues. You can take neighboring residues. Here we use different window lengths, we can start from 3, right, 3, 5, 7. So, you can up to you can change the window length and see to which window length you can get the best fit. So, you can optimize the window length depending upon your problem. How to include the structural information?
Change in property.
Right, you can see the central residue. So, from the central residue you can make a sphere and account the residues which are occurring within the limit and take the average property values. This space you can change, that you can say from 4 angstrom to 14 angstrom with different intervals, say 1 angstrom or 0.5 angstrom, and you can optimize this space to account the stability of these particular mutations. Essentially, if you see around 68 angstrom is sufficient to accommodate the medium range interactions and the long range interactions. It may work well to understand or to account the stability upon mutations, right, in terms of the surrounding residues plus the mutant residues, right, we can do that. So, we can relate amino acid properties and the stability. So, for this will give you which type of information, right, you can understand okay, which properties are important and depending upon the mutation type, whether it is a buried mutation or exposed mutation, is helix, strand or coil.